Hello, my name is Shui Ma, and I'm an assistant professor at Seattle Children's Research Institute. Today, I'd like to share with you a recently published collaboration with David Sherman at the University of Washington, Christoph Grudner at Seattle Children's, and Q Ree at Cornell University, focused on identifying Mycobacterium tuberculosis transcriptional regulators that alter its viability under different stress conditions. This project is motivated by the need to identify novel drug targets to treat TB by understanding what molecular factors in the bacteria control its infection outcome and treatment response. We know that much of TB's response to dynamic host and drug exposure stresses is coordinated through its molecular networks. These networks and their regulatory controls constitutes a powerful but underexplored window into novel drug targets and treatment shortening strategies. Therefore, we developed an approach that could systematically identify regulatory network elements that are critical for context-dependent TB survival. With Tig Rustad in the David Sherman lab, we developed a forward genetic screening approach called Transcriptional Regulatory-Induced Phenotype, or TRIP screen, which quantifies the effect of inducing each TB transcription factor on bacterial viability. This TRIP screen is built upon a comprehensive library of transcription factor induction strains, each of which can induce the expression of a different TB transcription factor upon exposure to chemical trigger. These strains can be pooled and grown as a single culture, either in the presence or absence of the induction of expression. In the presence of induction, strains expressing regulators that benefit fitness will become overrepresented in the population and will have increased relative abundance in the pool. Strains expressing regulators that are deleterious to fitness will become depleted in the population and will have decreased relative abundance in the pool. We can quantify the relative abundance of each transcription factor strain by sequencing regulator-specific barcodes from the strains and use this as a readout for regulatory fitness. Once specific regulators are identified, we can use previous profiles of the regulatory network, mapping where the regulators bind and what proximal expression changes the regulators elicit, in order to identify candidate effector genes responsible for the phenotypes. The TRIP screen offers multiple advantages relative to other genetic screening strategies, such as T-and-seq. Whereas T-and-seq relies on individual gene disruptions to identify essential genes, TRIP links fitness to regulators and the regulatory subnetworks, gene sets that were co-evolved to coordinately express together. This enables identification of multi-gene emergent phenotypes. In addition, the TRIP screen requires a much smaller number of strains to achieve comprehensive coverage of the regulatory network, and this enables screening in a broader range of conditions. Together, TRIP and T-and-seq represent highly complementary screening strategies for linking genes to phenotypes. In this study, we focused on quantifying the TB regulatory fitness phenotypes under log phase broth culture growing conditions and under treatment with the anti-TB drug isoniazid. We can quantify the fitness for each transcription factor under each condition as the relative abundance fold change of the corresponding strain in the presence versus the absence of expression. The resulting score is negative for fitness deleterious strains and positive for fitness beneficial strains. When we plot the TRIP profiles and the untreated growth on the x-axis versus the isoniazid treated growth on the y-axis, we find several groups of regulators emerge. Most transcription factor induction strains do not have significant differences in abundance fold change, either in the absence or the presence of the drug, indicating that regulator induction has no impact on fitness. This has been confirmed in growth experiments in monoculture, such as in this representative example regulator. Some transcription factor strains show significantly reduced abundance, even in the absence of drug, indicating that their corresponding regulator's expression conveys growth defect, such as this representative example regulator. Others show significantly differential abundance during isoniazid exposure, indicating that their corresponding regulator conveys isoniazid tolerance or isoniazid hypersusceptibility. Among the regulators that conferred hypersusceptibility was MCE3R, previously implicated in regulating lipid transport in TB. We wish to investigate its role in isoniazid susceptibility further. In monoculture validation experiments, we found that inducing MCE3R significantly increased isoniazid killing at each time point measured, and that induction also potentiated isoniazid-mediated bacterial inhibition at every dose that we tested. To investigate the mechanism of MCE3R-mediated isoniazid hypersusceptibility, we compared the genes that differentially express upon MCE3R induction with the genes that the wild-type TB differentially expresses during isoniazid exposure. We found one gene, CTPD, which is upregulated in the wild-type response to isoniazid and repressed by MCE3R induction. We hypothesized that MCE3R-mediated isoniazid hypersusceptibility could occur in part because of this conflict in CTPD regulation, and thus that disrupting CTPD independently should also convey isoniazid hypersusceptibility. Validation experiments with a CTPD transposon disruption strain showed significant survival defect during isoniazid treatment, consistent with our hypothesis. While CTPD had not been previously implicated in isoniazid response, it has been found to have an effect on efflux of iron from TB. Given that iron plays a role in regulating the conversion of isoniazid prodrug to its active form within TB, we collaborated with Q-Re and Vijay Soni to measure levels of the activated form of isoniazid in the bacteria. We found that CTPD disruption results in significantly elevated levels of the active form of isoniazid in TB lysate, which could explain CTPD's role in isoniazid susceptibility. In summary, we have developed TRIP, a novel forward genetic screen that reveals regulators and the regulatory subnetworks that confer fitness phenotypes under different stress conditions. We use TRIP and regulatory network analysis to identify a novel role for the iron transporter CTPD in influencing isoniazid susceptibility by modulating levels of the activated form of the drug. Our case study demonstrates the power of this approach to uncover effector mechanisms of other regulatory network phenotypes detected by TRIP. Our next steps will be to harness TRIP to study TB regulation of infection fate and treatment outcome in a host context, as well as to adapt the strategy to study TB signal transduction in collaboration with Christoph Grüdner. To close, I'd like to thank the many collaborators who made this possible. This project was completed while I was a postdoc in the David Sherman lab, and I'd like to thank all of my former lab mates for their support and experimental help in this project. I'd also like to thank the support of our collaborators in the Re and Grunter labs, as well as our funding. Thank you for your attention.